Competition or inhibition ELISA is the most complex type of ELISA method. इस मेथड में हम एंटीजन या एंटीबॉडीज़ की कंसंट्रेशन डिटरमिन करते हैं by interfering the process, means by interfering the antigen antibody binding. Therefore it is also known as inhibition ELISA. इस मेथड को अगर पहले से डिस्कस मेथड के साथ कंपेयर किया जाए, उन मेथड्स में हमने बात की कि एंटीजन प्रेजेंट है, एंटीबॉडी ऐड की, इफ़ दे आर कॉम्प्लीमेंट्री दे आर सिमिलर, दे कैन बाइंड विद ईच अदर एंड दे कैन मेक द कॉम्प्लेक्स. और उस कॉम्प्लेक्स फॉर्मेशन की बेस के ऊपर हमने कलर चेंज की बेस के ऊपर उनकी कंसंट्रेशन डिटरमिन कर ली. But in this method we are going to interfere this binding and then we are going to determine whether the particular antigen or antibody is present or not. Their principle can be the similar. For the principle for this method can be the similar as discussed earlier with the other formats like direct ELISA, indirect ELISA or sandwich method. And this method is often used when only one antibody is available for any particular antigen, or the sample which is available is very small. If the sample is very small, then there is likely a possibility इस sample को अगर हम बाकी दूसरे methods को use करते हुए detect करने की कोशिश करें तो हो सकता है रीजनेबल सिग्नल हमें ना मिले, और रीजनेबल सिग्नल ना मिलने की वजह से हम इसके बारे में जो इन्फॉर्मेशन है वो गैदर ना कर सकें. इसलिए हम एक कॉम्प्लेक्स मेथड यूज़ करते हैं इफ़ द सैंपल इज स्मॉल और इफ़ ओनली वन एंटीबॉडी इज अवेलेबल. जो स्टेप्स हैं टू परफॉर्म दिस मेथड इज सिमिलर. दैट first we add the antigen, or we coat the the plate, then we add the sample. After coating, blocking, washing steps, we proceed further to see if the signal is present or absent. अब इसमें दो चीज़ें पैरेलल देखने वाली हैं. एक हमारे पास जो कोटेड एंटीजन है वेल में, और एक हमारे पास फ्री एंटीजन है जो कि हमारे सैंपल में है. व्हेन वी ऐड सैंपल इन द वेल एंड बोथ द एंटीजन्स आर सिमिलर, जो कि बेस कर रही है, कोट कर रही है वेल को, और जो कि फ्री एंटीजन है, इफ़ दे आर सिमिलर देन देयर विल बी अ कॉम्पिटिशन फॉर बाइंडिंग विद द एंटीबॉडी. If sample antigen is high, that is free antigen is high, it will bind with the antibody and it will not bind with the antigen which is coating the well. And similarly, if the free antigen is less, then more and more antibody will bind with the coated antigen, and that gives us strong color. Free antigen antibody complex during washing steps is going to be removed. So increased intensity of the color versus decreased intensity of the color, that will tell us whether the antigen will be more in the sample or less in the sample. High sample antigen concentration will give us low signal output. Small or little sample concentration will give us high color intensity because of the reason that most of the antibody has bound with the antigen that is coating the well. This diagram briefly shows that inhibition or competition ELISA works on what principle. This particular picture shows you that we have this coated antigen which is coating the well, Then we added the sample in this well. This sample has a similar antigen, which is now free antigen or freely moving antigen in the liquid phase. Then we add the antibody. This is a primary antibody, and this antibody is meant for both the free antigen or the coated antigen. So depending upon the concentration of free antigen, this antibody is going to bind with the free antigen or with the coated antigen. For a moment, we suppose that there is high concentration of the antigen in the sample, so there is high concentration of freely available antigen. The antibody is going to bind with the freely available antigen. Then we do the washing steps. In this washing step, all free antigen antibody complex is going to washed off. Very less antigen antibody complex is remained back in the well. So the color intensity which is going to appear is low. The interpretations of the results by this method are opposite as compared to previously discussed ELISA method. One is actually measuring antigen concentration by observing the extent of signal reduction. जितना सिग्नल reduced होगा उतनी sample में antigen ज़्यादा होगी. And similarly, this method can be a direct method or indirect method depending upon if the detection antibody, which was also known as primary antibody, is labeled with the enzyme, or we need an other antibody, the secondary antibody, which is coated or which is conjugated with the enzyme. This method can also be of two types, that is direct or indirect method.